A very warm welcome to those who have just joined the webinar. We are waiting for everyone to join. We'll be starting with the webinar in 10 more minutes, and it would be nice if you could introduce yourself in the chat box and tell us about your educational background, your research interest, and your motivation to join the program. We're looking forward to listen to your introductions. Thank you. Hello, everyone. I would like to welcome those who have just joined. We'll be starting the session in more 3-4 minutes. We are waiting for others to join as well. Thank you for joining. And you can take out this time to introduce yourself in the chat box. Hello, everyone. A very warm good evening, good morning, or good afternoon, in whichever part of the world you are. I would like to welcome you all to today's free webinar on data science for biomedical research. So with the rapid growth of high throughput data, including omics technologies, gave rise to significant demand for data science skills and experience with bioinformatics methods of analysis. This online training program will cover the practical and conceptual aspects of data science, including data wrangling, statistical analysis, and machine learning in application of high throughput biomedical omics data using big data analysis tools on the T-BioInfo Platform, R, and Python. Throughout the course, the students will get an understanding of opportunities and limitations of machine learning in the context of basic preclinical and clinical research. The training is designed as a combination of online resources, practical hands-on experience, assignments, and live workshops that will be conducted via Zoom. And throughout the course, we will review several project examples that demonstrate the successes and limitations of conventional machine learning methods and deep learning using data from public repositories. So let me quickly introduce myself. I am Sonalika Ray, and I'm the Omics Logic Community Manager responsible for the interaction with thousands of our users that are a part of our bioinformatics community, which is managed by Biomedical. So before we get started with today's webinar, I would like to take out a few minutes to introduce our company. We are working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The training has been completed by over 24,000 participants from 187 countries in over 300 workshops, and due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers. This training has been completed by participants in 6 different specialization tracks, which include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. All right, so that was about us. And now I would like to highlight some of the current challenges and limitations and how we at Omics Logic help resolve these with our training and research services. So what are the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large and take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but do not receive practical hands-on experience with the full workflow due to the limitations that I just mentioned previously. Similarly, faculty faces challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they're unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about metagenomics of seawater. So what is the learning process? In order to create a clear path of skill development, Omics Logic employs the Bloom's taxonomy levels of learning. This approach starts with the basics, where the students recall the facts and terminology and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The students will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do Omics Logic resources help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics, composed of courses designed to take you through the theoretical knowledge. Omics Logic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give students a clear path of the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, an introduction to basic coding languages like R and Python, or beginning a data analysis project. The users will be given the directions to access the T-BioInfo Platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. The different stages of analysis are performed in different sections of this multi-omics platform. This program comes with an access to the Omics Logic courses, which includes various courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python, and machine learning for biomedical data. And example projects access of different fields which you could learn from and replicate for your research work as well. So first of all, let's begin by signing up on the Omics Logic Learn portal. I'll be shortly taking you to the portal and give a quick demonstration of how you can create an account on the portal and leverage the resources that I just mentioned. And once you have your account set up, please make sure that you have a complete profile, which includes a profile image, full name with appropriate capitalization, link to your social media handles, which could be Facebook, LinkedIn, Instagram, Orchid, or Twitter, and a brief bio about yourself that will consist of educational background and research interest. The mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of the course, the certificates will appear under the Certificates tab. Now I'll quickly change my screen and open up the Omics Logic Learn portal. Those who already have an account on the portal, please put a yes or one maybe in the chat box, and others can take out this time to create an account for themselves. Today, we'll be going through some of the exercises in R on the portal itself, and having an account would help you follow the mentor's instructions and also practice it alongside. So please make sure that you have an active account. I can give you minutes for you to have an account set up for you, and then we may begin with the webinar. So this is a quick demonstration for all of you. Click on the login button, and this will give you some options to either create an account for yourself or sign in with any one of your social media accounts. I just clicked on my Google account, and I would be using one of the accounts to get into my account. So this is the easiest way how you can create an account for yourself on the Omics Logic Learn portal. It's free of cost, and you can then explore the various courses and training programs that you can get enrolled into to leverage the resources. And yes, I'm awaiting responses in the chat box. Actually, if you have an account or if you are creating one and done with it, please put a 1 so we can proceed ahead. Yeah, I see a response. Thank you. We can wait for a minute or two for the responses and take this time out to sign up so that we are all set to practice the assignments today. All right, thank you for some of the responses in the chat. I hope others are creating an account for themselves as well. And we hope to have this session interactive today where we could listen from you the answers to the exercises that the mentors would be going through. So please stay attentive and also put in the replies in the chat. We will be awaiting them. All right, so once you have your account set up, please click on this email address, which says welcome back with your email address. When you click on it, you will be redirected to the profile section. Now, this is a portrayal of your achievements that will help the mentor know about your progress, and also the recruiters look at your achievement. So please make sure that your profile is completed and you have all the fields filled in appropriately. And this is the way how we can monitor your progress. And then all the courses that you would be completing as a part of this program, you can look at the progress or the completion on the profile tab itself. And once it's completed, you can download the certificate from here. All right, so that was about getting started and having your account on the portal. Now we are all set to get started with today's webinar, and I would now like to pass on the stage to Mr. Ilya Brodsky to give us an introduction to the program. Over to you, Ilya.
Hello, everyone, and welcome to this session. I will briefly introduce some of the objectives of this training, and then I will pass it on to Dr. Raghav to speak about how this will be done and what are some examples of things that you could do with this. So the data science program is designed to introduce statistical analysis and machine learning, provide you with an understanding of how data is generated. So how you can, for example, think about your experiments if you're going to be using data science to analyze your own data, or how to evaluate other people's data and spot trends that might represent technical versus biological variation. We'll talk about the significance of data mining and finding patterns that can lead to interpretation. So you can actually think about patterns that you identify, complex patterns that are not simply, let's say, differentiation between means of groups, but how to find these complex patterns and interpret their association. And then finally, how to study complex biological systems using big data. And so when I speak about complex biological systems, any system in our body, for example, whether it is our nervous system or immune system, these systems are complex because they represent many different cell types, they represent many different associations and connections, and they are also dynamic. So there's a time course component in them as well. And so when we look at data, how can we apply data science and machine learning and statistical analysis to be able to actually understand these biological systems? That is going to be the major objective of this program. So how are we going to do this? First of all, this is going to help you understand various types of omics data and understand how data analysis that you see in publications, for example, are actually done, and how they are representative of both the biology and the data side of things. You'll talk about, you'll learn about the significance of exploratory analysis and actually how to prepare your data for, let's say, classification, how to prepare your data for biological interpretation. You'll be able to find patterns that are biologically explainable. And importantly, you'll learn how to communicate your findings to your peers. So you will be able to connect the dots between data preparation, data processing, data analysis, and biological significance. And I think the major objective here for most of the participants that are joining today is to really understand, well, how am I going to get to this where I am at, right? So maybe you are thinking, okay, these are some nice objectives, but how do I actually know that I am at the right level, that I have the prerequisites, that I'm ready for something like this? Well, that's exactly the goal of omics logic. Our goal is to actually be able to show you how anyone, whether you're a high school student, an undergraduate student, a postgraduate student, maybe a faculty, and still you can benefit from this program. So I'm going to take a few slides to speak about some of the important aspects of data science in general, and then speak about specifically how you can use the specific illustrations, case studies, and project examples that we will use in the full program to be able to understand and achieve something with this kind of training. So first of all, today I'll talk about some of the specifics for biologists, right? We'll talk about data science, but specifically we're talking about data science for biological data. And I'll talk about some of the challenges and some of the opportunities for someone who is coming from a biological background. And I'll try to make sure that the logic for why you would need to study these things, why you would need to use these tools, how do you actually go from A to B, and what would be the outcome. And so the goal of this whole training actually is going to be able to find and understand what data represents, how to prepare the data for analysis, learn code syntax, and be able to identify what is the question and how to answer it. And I think to understand why data science, it's important to understand what data science actually is. And that will help us kind of get back to that same point in a few slides and think about whether this is achievable. So data science, as you know, is the intersection between data, statistics, and some specialization domain. And so when we talk about statistics, typically we're referring to the balance between signal and noise in our data. Sometimes we can, for example, do some experiment, and maybe every once in a while we see some interesting pattern. Maybe the gene that we are interested in sometimes is expressed, sometimes is not expressed. And what we would like to know is whether we can generalize our understanding, whether what we see in our experiment could then be applied to biology in general. And so that's where statistics can help us. It can help us understand how random patterns are different from patterns that are reproducible, recurring, and significant. And so that is the difference between signal and noise. And to be able to spot signal and noise, we either need a lot of samples or we need to know some advanced methods that can extract those specific signals and help us identify the ones that are reliable and the ones that are not. Then on the data, we have, in general, just the problem of noisy data and structured and unstructured data. Data is just not ready for the type of analysis that we would like to do. And this is especially true for complex data analysis. So when we think about machine learning, we typically think about data that has classes, that has many groups. Those groups are well defined. We don't have any failed processes because our data misses some numbers or something like this. And so that is really the problem with data. And then, of course, it's not important just to have some data. It's important to understand that data. And so when we speak about specialization, we can think about our understanding of that domain in terms of the terminology, in terms of the expected variability, and in terms of anticipated artifacts that we can try to identify, prevent, and avoid. And so that really brings us to a very similar chart that represents here bioinformatics. So today, bioinformatics is less focused on maybe some niche applications of algorithms and computer science, and in general is becoming broader because data science applied to any kind of biological challenge or problem or question ends up using that specialization domain of biology. And so what are some things that we need to know in biology that are important for us to be able to work with data? Well, one of the things that we need to understand is how this data represents biological, for example, phenomena, how this experiment produces data that I know operates with certain elements and I understand those elements. So that specialization domain is really something that we need to think about as we approach this topic of data science in bioinformatics. As you can see, bioinformatics and data science are very similar. The difference between them is that bioinformatics is very focused on biological problems, and data science is focused on general application of these tools. And somewhere in the middle of both of these sits the term machine learning, right? So we actually want to incorporate machine learning because machine learning, when it is properly applied to the right kind of question, to the right kind of data, and is interpretable, makes it useful for biological and biomedical discovery. And so today we'll briefly talk about some of these tools like supervised and unsupervised learning, and speak about some of the differences between deep learning and machine learning as well. But let's first turn to data. So as you know, biomedical research is an engine that keeps on churning out new data sets all the time. So you can see this graph on the left. You can see that over the 27 years that NCBI has been around, there has been an exponential growth in the availability of data that actually is available for all of us. One of the examples is this current pandemic that we can see has produced such a tremendous amount of data that today, when you speak about mutations, variants, sequencing, everyone knows these terms and not just people who specialize in that domain. And that just shows you not only an example of how data is generated, but also the trajectory, how this data will continue to be generated also in the future. Now, the problem is that most of this data that is being studied is actually unstructured. And so if we think about this process of data being generated, and then how this data needs to be organized so that we can start mining it for patterns, we then understand that the process from raw data to actual insights and therefore action leads through a pipeline of data scientists, right? And so as we become more informed with data science, more skilled in data science methods, we now can participate in translating this raw data into action. And to do that, we need to understand the different types of data, and we need to be able to identify and learn how to use data that is already structured. So what is, in plain terms, the difference between structured and unstructured data? Well, if I take a survey, I can ask you, what is your level of education? And many of you will have different answers. Somebody will say, I'm a graduate student. Someone will say, I am learning biology at University XYZ. Somebody will say, I'm a professor. So how do I analyze such a diverse set of answers? What I ultimately want to do is categorize everything that I have into just a few small groups and compare them between each other, similar to what I see here on the right. And that is really the difference. So how do I go from this unstructured stream of information being shared in different formats to ultimately something that is organized and easily comparable? Now, this is a simple example, but let's think about biology. Well, in biology, a lot of times we're dealing with high throughput automated technologies. These are technologies that can produce images, they can produce sequencing data like genomics, epigenomic peaks that I can study for different kind of variation produced by epigenetic processes, or I can analyze gene expression tables. All of these different types of data come in an unstructured format. And the first goal that I have is to actually organize them into a table, into a matrix, because a matrix is something that now I can apply different types of data methods, data analysis methods to. So all of these types of data, imaging, clinical, streaming data, omics data, chemical data, phenotypic data, what first we will need to do is to organize them as structured data. And once we have a lot of such data, now we can connect the dots between patterns and their biological interpretation. And so a major portion of what we will do in this program is link all of these steps together so that we have a coherent plan. We know how to approach these kinds of problems. We know what each data type represents, and we know how to get from raw to structured to interpretable insights. So Narika, if you could please monitor the participants in terms of sound. That would be great. So ultimately, our goal is in the end to be able to ask questions, identify data sets, and produce answers that are reliable and can be used for actionable activities. So we'll talk about examples in basic research, translational research in biotechnology and pharmaceutical industry, as well as biomedical applications such as clinical trials and precision medicine. Ultimately, the goal, or what you should expect to have after you complete this program, is number one, how to find data and understand what it represents. So we'll actually use a few case studies, examples that we will provide for you, but we will also guide you on how to find similar data sets on public repositories such as NCBI and others. We'll also understand how to prepare data for analysis. So we'll talk about how this raw data could be downloaded, or what are some effective tools for you to be able to use on the cloud so you don't have to deal with huge amounts of data that this represents, and be able to process them and prepare them for analysis. You will also learn code syntax. So you understand not only how to read code, but which part of the code represents what type of method, and how to modify that code so that you can apply it to a new data set that you're interested in, as well as statistics and machine learning fundamentals. So that means that you can understand the principles. Why are we looking at p-values, at r-squared, at area under curve? What do these things actually mean? Then you will be able to look at a data problem and understand what is needed to solve that problem. So framing a problem correctly is very important, because you'll be able to start asking questions that could actually be answered with a process. And then you will be able to navigate biological or biomedical references to be able to also interpret and validate results of the analysis, because a lot of kind of questions to these machine learning methods is, well, what does this method really do? How do I interpret this? And how do I know that it's a valid interpretation? And finally, we'll also give you an opportunity to be able to communicate and contribute to data science and machine learning code of others, or communicate your project results to others as well. So really, Omics Logic provides you with a complete roadmap to data science, specifically in application to biomedical research data. And we hope that this kind of training can simplify the path in this complex field of biomedical research from where you are today to where you ultimately want to be, which is whether you want to advance your career, develop your first research project, or use this kind of methodology in your current research. With that, I would be glad to briefly mention that all of this content is supported by an asynchronous library of materials that you can refer to during the training. And this material is enriched by case studies in various domains designed to introduce the specific terminology, specific data sets, examples, and case studies that you can start with so that you can develop your own independent project with this kind of data in mind. With that, I am happy to answer any questions that you might have. And if you do not have any questions, you can also mention them in the chat, and I will be monitoring the chat for until the end of this session, and I'll be able to answer those as well. So with that, I'll pass this on to Dr. Raghavendran Lakshminarayanan to take over.
Thank you very much, Ilya. Let me find ways to share my screen and okay, great. My video is also working. So I am Raghavendran. I'll be one of the mentors for this data science program. And today, actually, I want to give you an introduction to the kind of analysis that we will be discussing in this program. Yes. So let me show you, or let me share you a pipeline that we have set up in the server T-BioInfo platform. Pipeline is actually designed to compare, contrast, and benchmark different clustering methods using gene expression data. So to give you a very short overview about gene expression data, we all know that this data contains vital information that is required to understand the processes that takes place in a biological organism in relationship to its phenotypical conditions or characteristics or environment. So to understand and deciphering these hidden patterns in this gene expression data will help us strengthen our understanding of the complexity that is buried in the biological networks, and the complexity, the general complexity of the gene expression data or omics data and the volume of the genes present increases our challenges of comprehending, interpreting, and analyzing these huge mass or volume of data. So the use of data mining techniques like clustering is one of the first few steps towards addressing these challenges. And we need to do, or we need to perform these analysis methods to process several patterns that are buried and interesting patterns that are underlying in the data. So let's go back to the pipeline, and I want to bring to your notice to the sequence of analysis that is involved in this pipeline. So we start with the principal component analysis in our library, and then we follow it up and we take the output of the principal component analysis and then apply a multitude of clustering techniques like hierarchical clustering, like k-means clustering, spectral clustering, DB scan, etc. And we finish we end the pipeline. So we do this to compare these clustering methods interpretation of the patterns within the gene expression data that was provided as an input. So before running to the output files, let me tell you or briefly show you what the input looks like. So if you check this input, this is an expression data, and this data will look like something like this as a table. This is a structured data that Ilya had introduced you to, and this data is organized in such a way that the samples are in the columns and the genes are in the rows. Usually this data will be of a huge dimension, right? The dimensions of the gene can vary anywhere between tens of thousands to more, even more, like 20s of thousands. So if you check this one, we have curated this data and we have around 7,000 genes in this data. Even this is big data, right? So to understand the patterns of this big data, what we actually do is we perform principal component analysis before clustering. So what PCA does is, PCA is actually used to project this data of huge dimensions into a lower dimensional subspace, and then the clustering is applied in this subspace. So using PCA before clustering actually logically reduces the or decreases the computational cost, as we are actually implementing clustering on the reduced dimensions of the data. PCA will actually provide distribution of the data points in the transformed dimensional space, but PCA itself will not cluster the data points. So to demonstrate you, let's explore some of the outputs that you find in the pipeline. Once you run such pipeline, you will get some visualization options. And if you take some of the visualization option, I think I have already loaded this visualization. So let's go there. Yeah, so you can explore the principal component analysis output of this pipeline in a 3D interactive plot. So this immediately shows us there is some really good separation between the samples of different types. And if I can move this over, yeah, and we have the color coded legend also available. So we quickly understand that there is a very well or good separation between the samples, or at least between the luminal and non-luminal type, and we can employ clustering to observe whether these patterns are actually in the clustering techniques or not, or do we have any? Do we see any other surprising information in the clustering? So if you check another output, which is the tabular output of this principal component analysis platform, then we will see that the dimension, which is of 7,000 genes that we have inputted, had been compressed to 3 here we have given only 3. It could be depending upon the setting, 3, 5, 10, or how many components that you are going to calculate, right? So thus a decrease or using clustering on this decreased dimensional space will actually speed up your process and also will eliminate the unwanted noise which could be present when you are dealing with the high dimensional data. So to understand all of this pipeline, we will have several of these examples in the session or in the program. But now I want to point you or focus on the bottleneck of this pipeline, which is the principal component analysis on our input data. So if you click on the output files, you will get outputs that are associated with all the analysis that we have run, we have actually used here, included here. And if you include or if you download this principal component analysis _r pack.zip, and this actually contain the code that was necessary for the principal component analysis to generate this output. So let's go there. Let's take it out. Yeah. So this pack contains the input file as we had downloaded and I showed it to you, and it contains the standard R code with which the server actually ran this analysis. So let's, yeah, this is the R code which the server used to run this principal component analysis pipeline. But we have given or we have dissected this R code in a step-by-step manner in an associated lesson, which you get a free chance or a chance to explore only for today. So if somebody can paste the link, let me find the link myself. Yes. So this is a lesson that deals with the principal component analysis of your input data. And I give you the link for this lesson in the chat box. You are free to open this lesson and explore the codes along with me. And we can walk through the different components of the code, and we will have a separate space where we are going to apply this code and run this code from the Learn Omics Logic platform themselves. Yes. Okay. So in this lesson, we start with an introduction to what principal component analysis can do and how it is very useful for us to first explore the data using principal component analysis as such. And after you have passed through that, we give some examples of the input methods or input table. Then we discuss this table now, and we will also discuss this table in greater detail during the program. And in the Learn the Code section, this lesson is organized by first giving you an introduction or a specific aim for what this lesson is intended to teach you or explain you. And then we straight away jump into Learn the Code section, where first you get a demonstration of how to use PCA for your data. And then we have a code breakdown. So what do we call as code breakdown? We break down the codes into fundamental components of the code and explain the relevance of this code and explain how it is or how different components of the code and the syntax of the code come together and help you perform this analysis. For example, we have to load some of the libraries which will enable you to run this analysis in, for example, in R's environment. So it is usually done by including the library function and followed by the name of the library. And then we definitely have to load our input file, and this is done by defining where the file is or defining the location of the file using this syntax. And this is the assignment operator in R, which will take the value of whatever is on this side of the operator and then assigns to the object that is on this side of the operator. And then we load up the table or load up the input file using a read.table function, and read.table function takes at least one parameter, and that one parameter has to be the location from where you can access the file, right? This location can be your local computer, or this location can be a publicly available link that we usually share with during these lessons. And we have to describe how our data looks like, right? At least for few of the parameters, we have to describe how our data looks like. If you check our input file, our input file contains, if I can go there. Yes, our input file contains the first row, which tells you what each and every column is supposed to be. So, or in other words, in programming languages, the first row is the header of the columns, right? We know that this column represents the expression value of the sample that is named by named this code, and so on and so forth. And we need to mention that to R so that the data is actually loaded correctly. So that becomes one of the description of our input file. And we have several other descriptions, like how two columns are separated. That will form the separator that separates two columns in our data. This separator can be a tab, this separator can be a comma, or this separator can be any special character that you can use, like semicolon, like pipe operator, and etc. And you can also mention some additional parameters, like how you want to treat the characters that is present in your data table, and whether or not you want to load the first column as a row name, which will be very useful for further purposes, right? And then once you have loaded the table into the object full underscore table, which in this case, the read.table function loads this as a data frame. So what is a data frame? You will learn much more about data frame and etc. In the sessions. So in a nutshell, data frame is a collection of data of various types. A matrix or an array is a collection of data of specific data types, but while data frame can have data of different data types, like a character, like the logical character, like categorical character, and numerical character, of course, numerical inputs, of course. So in order to run several of these statistical analysis and several of these analysis, we may need to convert our data frame into a matrix format so that the numbers are interpreted properly. So we do that by converting only those part of the data frame which contains the numerical information, and we convert that into matrix, and we store that into a matrix by the name expression. And then we have to apply principal component analysis along the genes as dimension. So if you see, or if you notice our input data had the genes across rows. So we need to transpose the table, and that's what this specific code does. And then we can, now our input file is ready and data file is ready to be analyzed for principal component analysis, and we can invoke, or we can call the application, or we can apply principal component analysis in R by calling the function prcomp. And again, prcomp at its basic level takes at least one input or one parameter that has to be the matrix that for which the principal component analysis has to be calculated. And we mention that matrix, and in addition to that, you can mention other options like how many components you have to calculate, or you can mention other components like do we have to scale the data, do we have to center the data, etc. And once you run this analysis, then you can store the result of this analysis in this object PCA. And there are several options in R that will leverage the different organizations of information that is present in this PCA object, so that we can plot, we can explore, and we can understand the principal component or analyzed components of our input data. So at the basic level, we can actually plot the principal component analysis first two components, right? PC1 and PC2, we can simply plot it, and we can understand that it produces some good separation of samples. Right? But this is just to see that whether we have any outlier in our sample, or how do we go about interpreting our data. So this is very basic plot, which gives us very little information, like for example, in this case, we see that there are no alarming outliers in our data, and there is some sort of grouping that is observed, but without labeling these groupings or without annotating these samples, we don't know what these where these samples are, what these samples come from. So that is what we can do in the following plots, where we explore different options, or exploring the range of data visualization options and parameters, so that you can label, you can color, and increase the amount of information that you can extract from this principal component analysis pipeline or output. So just like using plot, we have another way of interpreting the PCA, which takes GG45's library, and Autoplot is that function, and that function comes with added advantage of having some presets that are very useful to us. Like, it takes the percentage of variance explained by principal component 1 and principal component 2 automatically, without the necessary of explicitly defining there, and it produces some really nice appealing graphics. And in addition to that, what we can do, we can expand, as I mentioned, to other options or other parameters in this autoplot function to include the labels for the sample, and we can color the labels, and we can color the mention the size of the labels, and that will add one more layer of information that we can that will help us understand the pattern. Now we can see that how where individual samples are actually located in our principal component analysis. This is useful, very useful, if you have only a handful number of samples. Even in this case, we have something around 40 or 52 samples, and it becomes really difficult to read some of them and to understand what that sample means, or what this group means, or what this specific sample mean, and what are the characteristics or phenotypical characteristics or different biological conditions under which the measurement from these samples are taken. So we can actually annotate them based on the phenotypical characters, and so that they separate very well, or we can at least understand, do they separate very well according to phenotypical characters or different sample characteristics that we associate from the for the data, etc. So before that, we can also enable loadings, which will tell you what are the important dimensions, or what are the significant dimensions that contribute the most to separate the samples into these multiple groups. We observe that there are some groups that we can observe in these samples. And if you enable loading, we can see that there are some specific genes like this 160180 and 065618, and we can understand that these genes or these transcripts are responsible for isolating the samples to this group, and while these genes are responsible for isolating the samples to this group. And so hence we can gain more information, not about how or where the samples are grouped, but also what are the most informative genes in our dimensions or in our input table. So we can expand the principal component analysis not only to 2-dimension, but also to 3-dimension. In some of the cases, that will be very helpful, like how you interacted with the interactive 3D plot that I just demonstrated you. And in addition to that, as I mentioned, when you have annotation available, or when you have the sample characteristics available for your input table, we can load those sample characteristics also into a separate object, and we can use those sample characteristics to color the samples and color their distribution in the principal component analysis output, so that we can understand whether the PCA's variance explained actually separates the samples according to the different characteristics that we associate the samples with. We can extract these sample characteristics from biological condition under which the samples are measured, or different phenotypical characteristics of the sample as such. For example, if it's a gene expression data from tumor sample, then we can take whether these samples are from what subtypes of tumor, or whether these samples are from tumor samples or adjacent normal samples, and any other layer of molecular characterization or histological characterization has been performed on these samples, and whether we can associate that to this sample, or you can explore a little bit beyond those molecular characterization of the samples to the clinical characteristics of the sample, like the demographical distribution, like age group, like ethnicity, whether they are actually represented in the gene expression data distribution of our analysis, right? So how do we do that? So we actually now again go through some of the important codes that we already had some introduction to. So we load the packages. Some of the GG45 is one of the package that will let us use autoplot function that we have discussing so far. And in this read.table function, as I had told you, we specify the location of the sample here, and then we specify some of the characteristics that is associated with the sample, like the separator, like whether or not the first row mentions the header, and then we can add some associated options which will help us organize the data, like how characters to be interpreted, and whether or not do we have to check for duplicate row names, and etc. So if you load this data, and then if you check first few lines of the data, we can quickly find out that the first column is okay. The first column will actually give you the annotation of the sample, and we can extract only the numerical component of the sample by selectively by selecting those specific numbers of column that we want to extract, and then we can apply principal component analysis only on the numerical component of this column. And if you do not do that, then this analysis is going to throw you an error that we have a non-numerical character or non-numerical counterpart input in your data. And here we enable scaling to be true, and immediately again we can use some basic functionality like autoplot to plot it. And in the autoplot function, as I was mentioning in the above section, we can specify, or we can actually include the annotation or sample characteristics that is associated with each and every sample. We can color the sample according to their annotation that we have included in the input table. So in this case, we are going to plot the principal component analysis result using autoplot function, and we are going to mention where this annotation is coming from. It is coming from the data table data, and the column that gives you the annotation is group. And when we do that, we can immediately see that the samples are colored, and our analysis of this input data reveals that we have very good separation of different sample types, like the normal, like luminal, and all the 4 subtypes of this tumor. They are very well separated, as you can see, and that is very good, very nice to know. And we also find out that the principal component analysis actually covers quite a bit of our very good range, or explains a very good percentage of variance in the first 2 principal component themselves. Like the principal component 1 explains 50% of the variance that you find in the original data, and together they explain something around 68% of them. And in addition to that, again, we see that we can actually label them, and that is okay. We can actually label them according to the sample size, sample type, and we can also we can label them according to the sample types, and this label has to be sample type. There is some error, then I will take care of that. So once you learn about how you can perform different principal component analysis, and how various different visualization options will help you understand the output of this analysis, and you can fine-tune the output or fine-tune the visualization for your need, for your necessary. Maybe you need only up to this point, or maybe you need to include the sample types or sample labels along with this output. So you can do that with the given options that is available. So once you have learned this code, so the next area or the next section in this lesson will help you practice all the codes that you have learned in this practice and learn the output section. So in here, we give you a console, which is an R console by itself, and we include the codes that you have just learned in the previous sections. And we first give you the codes as such and explain it to you that you can simply run this code, and you are expected to understand, or you are expected to see some of the answers or some of the plots that is associated with this code that we have seen that it can produce in the learn the code section. So I simply run this code and see whether it runs or not. Yes, I see that the output tells the success in running the code, and the answer is correct. And I produce several plots, and I also produce an output of my principal component analysis variance. And I can open these files and see whether the output that I created agrees or goes very well, or described how it is be described in the learn the code section, because we are using the same input data. We can check that. Yes, okay, it does. Very good. So now you can start exploring different parts of this code and start fine-tuning and start changing some of the parts of this code so that you can expand your understanding of the code that you have learned so far. And some of the hints that we have given here, for example, if you see in this area where we are calculating the principal component analysis, we again have not used the option to scale or to center the data. So we can actually do that, and some of the example codes are given here. So you can try scaling and centering the data. You can now modify this by including scale dot equals true, and center equals true. And then if I run the code again, I can clear the console and the outputs by clearing it. And then if I let it clear, if I run the code again, okay, now I have changed something in the data. So this checks for the pre-calculated data. So by centering and scaling, some of the outputs have actually changed, but that's okay. That's why my pop-up said success in running the code, which is what my aim is, but the answer is incorrect because this code block is intended to produce some other answer. So now I can actually check whether or not I see a difference if I perform scaling and centering in my original data. In the principal component analysis. If you have saved this one, then you can easily check. And you can again come down and see how you can write the output. Right? I just showed you from the server how you can access the principal component analysis that is calculated for your input file, and you can save it as a table. So, you can use the table for further downstream analysis like clustering and other methods. So this code block actually tells you how to save the table, right? And a necessary piece of code or a focused piece of code is actually given here, where you can find out how to write an output from R's environment into a file. And again, we have to tell what to write or what object to write out, and what is a file name, and some associated characters, how this file name has to be, or how this file has to be created by, like, do we have to include column names? Do we have to include the row names? Or how two columns have to be separated, just like how you have described the data while loading in the data. So let's run this and see for ourselves. Yes, in addition to a couple of plots, I can also see there's a table that is produced, and you can open this table, and you can download this table, save it as download this table, and find out about this principal component analysis calculated for your input file. So let's time constraint, let's not do that. I will let you explore that. You can download this table, load up in Excel, and then explore this table further. And then after that, what we have actually done is now you got a good hang of how the lesson works and how you learned the code and how you can access or how you can use the R console to run these codes and even modify these codes to suit your necessary, suit your requirements, And now in this section, we have deleted some of the syntaxes to keep you interesting. And then you have to find out where we have deleted the syntaxes and complete the code there to run this code successfully. So if you run it as such, it's going to throw you an error and error running the code. And most of the time, like in any programming language, the error message that R is going to give you is going to be very difficult to understand. So we clear this, and then we can go through this code line by line. If you had understood, or you can also refer to what we have done here and what we have given here, and find where the necessary completion is required for this. And I see, okay, I see that the principal component analysis function is not given in its full form. And I also see that there is a problem in plotting the principal component analysis. So let's complete it, PCA res. And I can also see that you have to run it time and again and see if this produces any error or if it's not producing any error. Now I have rectified some of the portions, and there are some more portions that I need to find out where the error is. And one of the areas where the error is, it's here. And I have to mention which column corresponds to coloring or annotation in the data expression table. That is group. And if you can know that if you open it in another file or in another tab, you will know that. And let's see what else do we have. And we can actually see here, okay, in this plot, we have to mention, we have to enable label. And in this plot, we can mention, we have to disable framing. Okay, once you do that, we can again try running the code. Okay, this time I made all the necessary changes, and I get success in running the code, and the answer is correct. And I create a lot of plots, which is very what all the plots that we have actually explored, and that will help us understand the patterns in this data. So after this, after letting you complete the code, and we give you an assignment where again some of the parts are missing, and you can again explain or try running this. So I give you this as a challenge. So today you can try completing this one assignment, and let us know by email or by email if you can successfully finish this option or not. And for those who wants to know more or who wants to explore a little bit further than what we have learned so far, we have more options where we have another input table. My computer is frozen. Yes, we have another option for another input table, which will help you understand the power of principal component analysis in identifying outliers. So you can go beyond the basics here, where you will get a taste of what I have demonstrated you in the server. You take the output of the principal component analysis and then apply k-means clustering to the table to identify the different types of clusters or different number of clusters that your principal or your data contains, or your k-means clustering identifies in your data table. So yeah, with that, I would happily answer some of the queries that you have and hand it over back to Sonalika.
Thank you so much, Dr. Raghav. Such a wonderful session. If you have any questions, please put them in the chat box so that Dr. Raghav can answer them. All right, I am taking this as, okay, we have a question. So Dr. Sara is asking, which tool or software do you use for PCA? Dr. Raghav, would you like to expand on this?
Yes, I mean, you can use R, Python, and those are very popular methods to calculate PCA, right? But PCA is also calculated in other statistical softwares like SPSS and other ones, etc. But we would use R and Python because of the flexibility that it gives us and because of the visualization option that it provides us, right? It can tackle, or you can use R and Python to have any sorts of, or to analyze any type of data, and not limited by the framework of other statistical software, right? But almost all of the basic statistical analysis softwares will help you perform principal component analysis, which is one of the basic analysis or exploratory analysis that you can perform for your data.
All right, sir. Sarah, I hope that answers your question. And
I also
Kruti is
got
asking,
a direct question.
yes,
Yeah,
sir.
I also got a direct question from Kriti saying, where have you downloaded the data? So the link for the data is given in the associated resources themselves. So in the lesson that I walked you through, so there are 1, 2, or 3 links for some of the curated data that we are providing you so that you can understand analysis on omics data types. So that's where we got the data. And while throughout this program, you will find various of such, or you will get your hands on to variety of such curated data. And also we will explain it to you, or we will have sessions that discuss example projects, which is based on publications and their associated data. So you will have variety of ways to get different data and understand different data formats. Yes, Annalika, you had another question.
Yes, sir. Emmanuel is asking, what roles does data science play in biotechnology? Can you please explain with a practical example?
Okay, I think Ilya actually covered that part, but I will tell you one more time. So there is humongous amount of omics data type that is being generated and that is being submitted to the public repositories. And there is a huge mismatch between the amount of unstructured data and the structured analyzed data that one can observes in the public repositories and in publications associated with that, and etc. etc. So this is where data scientists will come into play, right? They can take these unstructured data, find patterns in this data, and find patterns that is relevant to answering some specific biological question, or you can just simply use this data from a data-centric point of view, start exploring the data, understand some basic pattern, and then devise a hypothesis, devise a problem, and then keep collecting data and explore further. So that biology as such is slowly starting to acquire some of the characteristics of data science because of the volume and because of the variety and because of several of the data that is generated by next-gen sequencing methods and of different molecular characteristics that one can actually perform from these biological samples. That will help us understand the levels from different components, like from DNA, RNA, proteomics, and metabolites and metagenomics, etc. So all of these will require complex data type or data analytics methods that one can easily employ.
Thank you, sir. I hope that answers your question, Emmanuel. And Partially is asking, what is the importance of identifying outliers?
Yes, I mean, if you do not, outlier can actually skew your data. Most of the time, when you are analyzing the data, you will be calculating, or you will be estimating the average, mean, or median of your data, and presence of outlier can definitely affect those estimates. And that is why it is absolutely necessary to not include or identify outliers in your data, so that you have you have solid analysis for your data, and statistical relevance can be more meaningful if you do not have, or if you do not include outliers in your data. I hope that that also answers your question. You can try it out by yourself. Like you can find the average of 1, 2, 3, 4, 5, 6, 9, 8, 9, 10 numbers and include one huge outlier like 1000, and that is not an average representative of the numerical data that you have. And if you exclude that average, you will find that you exclude that outlier, you'll find that the average is much more representative of the data distribution that you have. So,
right, sir. That answers Parsily's question. And one more question. Is it important to have a knowledge of R and Python to get started with this program, or a novice can get started as well?
We will start this program like how I described, right? We will start this program by explaining it to you, the logic behind such analysis. So you will start performing these analysis in Bioinformatics Platform so that you are not hampered by the necessity to completely understand R and Python's code. But we would recommend you to go to at least that far where you can go through these codes and take these codes and apply your data set or data for your code or some modified data for your code and get an understanding. So you will gain our help and a lot of resources for you to do that also. So our basic idea or basic aim is to help you understand the logic behind the problem or logic behind this analysis method. Once you have understood that, using different tool to perform the same analysis becomes equivalent, right? You can use a server, or you can use R, you can use Python. So all of these are different ways of achieving the same analysis method. So the aim is to help you understand the logic behind this analysis. And also we provide you, for those who are tech savvy or who are code savvy, we give you option of exploring the same in coding environment. And you can start exploring once you have gained so much confidence in understanding the logic, and you can slowly start running a couple of lines of codes in R and Python, and you will find it easy once you have gained an understanding of the logic behind this analysis.
Right, sir. Thank you so much for answering it in such a detail. And one of the questions that we usually get is about the sessions. So what would be the session timing? Would it be live? And what if the participants are not able to join the session because of their prior commitments, college classes, or their working hours? So the answer to that is that we'll be having the recordings of the sessions put on the program page as well. So if you're not able to join the live session, you can watch the recording anytime at your own pace and get back to the mentor on Skype group that we would be having for this program with your questions, and the mentors would be able to resolve it for you. So that way you stay in touch and make good progress with the program. And now I would like to quickly take you through the program page of Data Science Biomedical Research Program. When you visit the page, let me just quickly put the link in the chat for you all. All right. So when you're on the page, the first thing that you see is the schedule. So here we have mentioned all the session details, like when would be these sessions held, the dates, and the time would be 10 a.m. CST, 8:30 p.m. IST for the live session. But yes, again, we'll be having the recordings put on the program page as well, so you can watch the recordings at your own pace. The recordings would come up here once the session is complete. So here we have the session details, what topics would be covered. And along with each and every session, you need to go through some associated online resources, which are lessons from courses on the Omics Logic Learn portal, and you will get an access to these courses as a part of the program itself. And in these courses, you have the syntax as mentioned, as Dr. Raghav took you through one of these lessons. And then you have the practice session with inbuilt console where you practice the code block, submit the assignments, answer the quiz questions, and complete the lessons that way. Okay, so today was an introduction to the program. On August 19, the second session would be about machine learning and statistics. And then the third session, we'll be covering the unsupervised and supervised types of analysis. And in the fourth session of the program, we would take a look at different data visualization techniques, which will have the dimensionality reduction techniques and the other techniques as well. Then we'll highlight more on the unsupervised learning or clustering. This would be through running the pipelines on the T-BioInfo server as well and the code block. And then we will highlight supervised learning, classification. Next, we will dig more deep into feature selection and gene signature construction. I'm not going in much detail of the topics that would be covered in this session. You have the link with you and you can anytime explore all the topics that we would be covering. The next we go ahead with regression and generalized linear models, and then network analysis on September 13. And towards the end of the program, we're talking about selecting the machine learning methods for real world applications. So first you learn and then you learn how to apply it to the real world applications. And then you have a session on deep learning, its types and applications. And finally, you'll get a chance to meet the mentor to prepare your research proposal for working on a research project. So once you have the knowledge of each and every concept that is mentioned here, you can get started with your own independent research project that you can present as a poster in various symposiums or conferences, or even have a publication out of it. So that would be the last session where we'll be taking you through this. And this was about the schedule of the program. And if you want to enroll for the program, scroll a little bit more. And what you'll see are 3 different types of subscription levels. The first one is 45 days, and then you have a 60 days one, and then you have a 90 days of subscription. So the program length is of 45 days. Definitely, we'll complete the program in 45 days, like all the sessions would be covered till then. And we have mentioned what all would be covered, and you'll be getting an access to the educational pipelines for 45 days. If you want to work on a research proposal and have a poster presentation as an outcome of this particular program, then definitely you need like some good days after completion of the program to put these things up together and work on your research project. And then you have the 90 days of subscription that is for 3 months, where you will get the research license where you can explore data from these big data repositories, like download them and then work on them on the T-BioInfo server and run the code block as well, and have a full-fledged research project ready for publication. So this is about these 3 different levels, and it's up to you which level you would like to pick. And for students, we have some scholarships as well. So we will share the scholarship form with you. Those who are interested can reach out to marketing@omicslogic.com. And I'll just put thank you so much, Sparsh, for putting in the mail address. And those who are in India can reach out to us directly on our contact number that Sparsh will just put up so that you can reach out. And if you have any queries that you would like Sparsh to address, you can mail her and she will help you enroll in the program and get started with your journey in data science for biomedical research. That was it that I wanted to talk about. And if we have any questions, you can put in the chat box, or you can also unmute yourself and ask. I'll just give the access. Yes, now the participants are free to unmute themselves if you have any